Welcome everyone. Today we have the pleasure for interviewing Dr. Alexander Biederstädt from Technical University of Munich in Germany. Alex is a physician scientist and a group leader whose research focuses on developing novel adaptive cellular therapies for advanced hematologic malignancies. During his postdoctoral research in Rezvani Lab at MD Anderson Cancer Center, Alex pioneered in innovative approaches using CRISPR precision gene editing to engineer next-generation NK cell therapies. Today, we will discuss his recent paper published in Cancer Cell titled Genome-wide CRISPR screens identify critical targets to enhance CAR-NK cell antitumor potency. And on behalf of EBMT, Alex, thank you very much for accepting our invitation.
Well, thank you so much, Esra, for having me.
Yes. To kick off things, we hear a lot about T-cell therapies like CAR and CAR-T, but NK cells are gaining massive momentum. In your view, why are NK cells the next frontiers for cancer immunotherapy, and what makes them potentially superior to other immunotherapies?
Well, I think what fascinates me most about NK cells is that they really present a cellular therapy platform that has a clear potential for off-the-shelf therapy. So NK cells can be safely administered across HLA boundaries without inducing GVHD. And that really puts them in a very unique position as a scalable allogeneic off-the-shelf cellular therapy. And the early clinical trial data so far has really shown remarkable clinical activity in heavily pretreated patients, specifically with hematologic malignancies, while at the same time delivering an exceptional safety record with substantially lower rates of cytokine release syndrome and neurotoxicity, for instance, compared to autologous CAR T cells. That really puts them in a position to provide these safe and scalable cell therapy products that eventually might actually be deployed in a much broader context and potentially even in earlier lines of therapy.
That is really fascinating, and it sounds like we are entering a new phase of where engineering innate immunity becomes just as important as engineering T cells. And most CRISPR screening logic has been borrowed from T cell research. So why was it necessary to build a platform like precise specifically for NK cells?
Well, so going back to the previous thought, I think there has just been a considerable momentum in NK cell therapy over the last few years, and accompanying this growing clinical interest, there's a growing interest in understanding the fundamental NK cell biology. There's still some therapeutic barriers that need to be overcome. Namely, those are limited in vivo persistence, premature functional exhaustion, and vulnerability to the immunosuppressive tumor microenvironment. And previous studies had already shown that with the emerging CRISPR toolkit, targeted editing approaches can actually bias NK cell functions toward more potency and enhanced cellular fitness. However, by and large, the broader landscape of actionable gene targets in NK cells had previously remained unexplored. And this is exactly where our project started out. So we built a platform, we called it Precise, and the idea was to really create an unbiased discovery platform that is specifically tailored to NK cells and will allow us to systematically identify genetic programs that control NK cell fitness, resistance to exhaustion, and anti-tumor function.
That's really impressive. And what was the most surprising biological insights that emerged from your genome-wide CRISPR screens in primary NK cells then?
And for us, there were a couple of findings that stood out. So I think the first and most surprising insight was really the identification of these deeper cell state regulators that had previously not been implicated in NK cell biology before. So to just name some of the key findings of our study, like we were able to identify the mediator complex and the Cal5 ring ubiquitin ligase machinery as powerful determinants of NK cell potency. And these findings really highlighted, especially in the case of the Cal5 ring ubiquitin ligase machinery, a previously underappreciated role of post-translational modification in regulating NK cell function. So that was one part. The second aspect that was really surprising to us was that while the specific goal of the platform was actually to investigate NK cell biology, what we found was that there were NK cell context-specific hits, but we also found some degree of overlap with T-cell fitness programs. So suggesting that there might be evolutionarily conserved fitness programs across different lymphocyte lineages. So, and that has really large implications because it means that some of our findings might actually be transferable across and beyond NK cell immunotherapy. And then lastly, what also surprised us is that while we designed this platform really to identify negative regulators of NK cell function whose deletion can improve NK cell potency, but what we also found was that there was a number of positive NK cell fitness programs, essentially those programs that dropped out during our screen. And we looked at those programs in more detail and actually cross-referenced them to clinical data sets. And what we saw is that specifically those gene programs that we identified as positive regulators were exactly the same programs that systematically get lost in NK cells from cancer patients. So basically showing that, like, finding like a molecular basis and explanation why endogenous NK cell function deteriorates in the clinic. And that also has an interesting implication because beyond what our approach was mainly targeted to, which was the loss of function screening, this also opens the door to basically bringing back those positive regulators through gain-of-function technologies like CRISPR activation or CRISPR knock-in.
Yeah, very exciting findings. You combine these CRISPR-identified edits with interleukin-15 armored CAR-NK cells. How does the synergy between the cytokine armor and the genetic deletions work to overcome tumor resistance?
So to answer your question, I think like we have to separate two complementary barriers to therapeutic potency and cellular immunotherapy. And cytokine engineering and CRISPR gene editing, they can basically address these two layers in very unique ways. So cytokine engineering and especially IL-15 in the case of NK cells really drives NK cell survival, NK cell proliferation, and in vivo persistence. And we see that with clinical data showing that there's CAR-NK, IL-15 supported CAR-NK cells that remain detectable in patients up to 1 year post-infusion. So we see the power of IL-15 armoring. At the same time, cytokines, like by themselves, cannot fully overcome some of the intrinsic molecular programs that drive dysfunction under tumor challenge. So this is precisely where CRISPR editing then comes in and really allows us to remove these intracellular constraints by targeting specific pathways that basically control NK cell metabolic fitness, cytokine responsiveness, or resistance to the tumors, to the immunosuppressive TME. So I think to put it together, having NK cells that are both cytokine armored and CRISPR gene edited basically allows us to have intrinsically reprogrammed NK cells that are externally supported by these engineered cytokine signals. And together they can then result in stronger and more durable anti-tumor activity.
It is very powerful concept. As a general question, NK cells has shown some success in hematologic malignancies, but solid tumors remain still a challenge. So which specific solid cancer types do you believe will benefit from these enhanced NK cells most?
That's a great question. I think overall, like the field is very much moving toward solid tumors, and I think tackling solid tumors clearly represents the major next frontier for NK cell therapy and even beyond, like cellular immunotherapy as a whole. And in fact, our CRISPR discovery platform was specifically built around that clinical need. So our precise platform was essentially designed around a solid tumor rechallenge model to really capture this tumor-induced NK cell dysfunction and then identify regulators that are capable of overcoming those limitations. When we then went on to validate some of the findings that we were able to make, we actually tested them across a number of different difficult to treat solid cancer entities. So we tested them across pancreatic cancer, colorectal cancer, and breast cancer, just to name a few. And we really were able to see dramatic improvements in NK cell cytotoxicity across a number of different models. And just to name one very specific example, we were able to show that REH2 CCNC deleted TROP2 directed IL-15 armored NK cells were able to really improve NK cell function, not only in vitro, but also in vivo in an orthotopic pancreatic cancer model. So I think really to address your question, there isn't necessarily this one single solid tumor entity that really stands out. I think overall there are a number of challenges that need to be overcome, ranging from antigen heterogeneity to really like trafficking and infiltration barriers to then really modulating resistance against the suppressive TME. And I think this is really where NK engineering can do a large part to really overcome these barriers and get to effective cellular therapies for solid cancers.
Yeah, great. In your work, you also described PRECISE as a roadmap. So does this mean we are moving toward multiplex editing where we delete 3-4 genes at once to create super NK cells?
I think that's absolutely where the field is heading to. So I think there's definitely an increasing understanding and appreciation of multiplex gene editing, not only in NK cells, but also in T cells. And I think it makes a lot of sense and to really put together these rational combinations of different gene edits, having a synthetic target recognition machinery coupled with different CRISPR gene edits that can then generate these synthetic, more powerful immune effectors that are capable really to overcoming today's limitations in the clinic. Our CRISPR screening platform really helped to lay the foundation for these rational design approaches. So obviously to put together these rational design strategies, one first has to understand the fundamental biology. And this is really where our work provides a rich resource to understand like what specific pathways in NK cells shape NK cell function under stress. And obviously the logical next step, as you mentioned, is really putting combinations of edits together. And what we already saw in our own work was that the most powerful combinations basically were co-deletion of multiple different gene targets, specifically REH2 and CCNC. And we saw that there were synergistic and cooperative effects on NK cell fitness by driving in vivo expansion, cytokine responsiveness, activation, and increasing interferon gamma signaling. So there is really dramatic improvements that really are brought are identified in these combinatorial edits. So I think going forward, the goal really is to identify those combinations of edits that collectively then produce the greatest function improvement, but at the same time also maintaining safety and manufacturability of the product.
It really sounds like we are entering a new era of rational immune cell design where we can program multiple layers of function into a single therapeutic cell. As a last question, in 5-10 years, do you see CRISPR-edited NK cells becoming an off-the-shelf standard of care, could replace at least complement the traditional chemotherapy we are using currently?
Absolutely. I think genetically engineered NK cells definitely have a huge potential to become an important pillar of cancer immunotherapy in the next decade. And we are already seeing these very encouraging results in early phase clinical trials. And in fact, the field is really branching out, exploring different targets, different indications, even beyond oncology and autoimmune disease right now. So there is this huge potential. And I think this really comes back to the fundamental properties of NK cells that really provide this unique scalability and off-the-shelf potential that really is specifically helpful to reduce costs, increase access, and really offer solutions to patients that are in need of immediate therapy. So to your question specifically, I think rather than really replacing chemotherapy, I think NK cell therapy will actually be integrated into existing multimodal strategies. I think it will increasingly move up into earlier lines of treatment. We now see NK cell therapy achieving remarkable response rates in very heavily pretreated patients, some of which have already received autologous CAR-T products. So I think NK cell therapy is really unique in that way, that it has that can achieve these deep durable responses of a cell therapy product, but at the same time has the rapid availability of a biologic. So we're going to see how, due to its safety advantages, it will probably move into earlier lines of treatment, will become more broadly accessible. And I think the key next step for the field is really how to bring and deliver those same successes that we have seen in the early clinical results with hematologic malignancies, to really translate them into the solid tumor space, to really try to deliver potent NK cell immunotherapy approaches to the large number of patients which have solid tumors and are in urgent need of better treatments.
It's an incredibly exciting vision for the field, and your work is clearly helping to lay scientific foundation for the future. Alex, thank you very much again for sharing this insight with us today, and congratulations on your outstanding work. We look forward to seeing how these discoveries translate into next generation of cancer immunotherapies. Thank you.
Thank you so much, Esra. Thank you so much for having me. It was a pleasure. See you. Bye-bye.
Bye-bye.